Welcome to all students in our ROSE project conducted by Rayat Shikshan Sanstha Satara. I am Mrs. Shinde SR from Sadguru Gadge Maharaj College, Karad. We are going to study the topic biomolecules. In this topic, today we are going to study up on the enzymes and nucleic acids. Now see the first enzymes. The enzymes are the proteins and which acts as the biological catalyst. In our body, there are the large number of biological reactions takes place. And these reactions takes place in our body at pH of 7.4 and body temperature is the 37 degrees Celsius. At that temperature and pH, the biological chemical reaction take place in our body with the help of this biological catalyst and which is called as the enzymes. That means enzymes are the biological catalyst and which are the proteins. Every living cell contains at least 1000 different enzymes. For example, insulin, which is secreted by pancreas and which controls the blood sugar in our body. Also amylase, which is present in the saliva, hydrolyzes the starch. There are the different enzymes which are present in our body. Now see the mechanism of this enzyme catalyst, how these enzymes are catalyze the reaction. And this type of the mechanism is the lock and key mechanism. This mechanism is called as the lock and key mechanism. See in this mechanism there is the enzyme and this enzyme have the active site. The enzymes have the active sites and these are the substrate. These substrate are placed in this active site. That's therefore these substrates which have the right shape and size of this active site. And when these substrate are bind with this active site. See in this diagram, these two amino acids which are the substrate. This is the enzyme which have this type of the active site, and then these two substrate which are bind with this active site. And these enzymes changes the shape to hold substrate in proper position. These enzymes change its shape to hold this substrate in proper position. Then next, these two substrate, these are reacts with each other. Energy is absorbed and bond is formed between these two amino acids. That means the internal rearrangement leading to catalysis occurs. The reaction take place in these two amino acids. Here energy is absorbed and bond is formed with the help of this enzyme which acts as a catalyst. And in this reaction the water molecule is released and bond between these two amino acids take place which is called as the peptide bond. Then this product is released. This means this is the product and these amino acids are the substrate. Then they react with each other with the help of these enzymes and the product which obtained is the dipeptide. And again the product is released and enzyme returns to original shape. See here, enzymes returns in its original shape and is available to catalyze another reaction. Then another reaction again take place. In this way, the rate of this reaction is very high. The enzymes are so efficient that one enzyme molecule can catalyze the reaction of 10,000 substrate molecules in 1 second. That means one enzyme catalyze the 10,000 substrate molecules in 1 second. Therefore the rate of this reaction is the very high. In this way, there is the mechanism of enzymes is the lock and key mechanism because this shape is the substrate have the proper shape and size of this active site. Then only the reaction take place. Now see the industrial applications of enzyme catalysis, which are the conversion of glucose to sweet tasting fructose using glucose isomerase. This is the enzyme. This convert the glucose into fructose. Then manufactures of new antibiotics using penicillin G acylase. These are the enzymes. Also, the next application is the manufacture of laundry detergent using protease. Then manufacture of esters used in the cosmetic, which are manufactured by using the genetically engineered enzymes. In this way, different enzymes are applicable in the industries. Now see the next point, nucleic acid. There are the two nucleic acids, DNA and RNA. DNA means deoxyribonucleic acids, and RNA, which is the ribonucleic acids. Then what is the role of DNA and RNA? The DNA, which stores the genetic information, and RNA, which use in proper synthesis for putting genetic information. That is, the genetic information transferred to new cell by nucleic acids. The genetic information transferred to new cell by nucleic acids. Then DNA, which is found in the nuclei of living cells, and RNA found in the fluid of living cells. The source of DNA, which are found in the nuclei of the living cells, and RNA found in the fluid of living cells. Now see the nucleotides. That means the nucleic acids are unbranched polymers of repeating monomers, which is called as the nucleotides. The nucleic acids are the polymers, and their repeating monomer, which is present in the polymer of the nucleic acids, are called as the nucleotides. The nucleic acids are polymer of the nucleotides. And now see what are the nucleotides. These nucleotides monomers consist of three components. The nucleotide consists of three components. One is the monosaccharides, then nitrogen containing base, and phosphate group. Nucleotide consists of the monosaccharides, nitrogen containing base, and phosphate group. The monosaccharides, which containing the five-membered ring, which is the furanones. Monosaccharides, that means nucleotides containing the monosaccharides, which is the five-membered ring, called as the furanones. Then nitrogen containing base. There are the two types of the base, which is the pyrimidine and purine. Two types of the nitrogen containing bases. One type is the pyrimidine, and second type is the purines, and third is the phosphate group. In this way, nucleotides contains the monosaccharides, nitrogen containing base, and phosphate group. Now see which type of the monosaccharides are present in the nucleotides. There is the five-membered ring containing one oxygen atom, is called as the furanose ring. This is the furanose ring. And this type of the monosaccharide is called as the 2-deoxyribose sugar. The 2-deoxyribose sugar, which is present in the DNA. DNA containing the 2-deoxyribose sugar. In 2-deoxy, that means the carbon number 2 containing 2 H atoms. The oxygen atom is reduced from here, and therefore it is the 2-deoxyribose sugar. And in RNA, there is the presence of the monosaccharides are the D-ribose sugar. D-ribose sugar, that means the carbon number 2 having the OH group. And in DNA, only the carbon number 2 is the hydrogen atom. two hydrogen atoms. In this way, the sugars which are present in DNA is the 2-deoxyribose sugar, and sugar which is present in the RNA is the D-ribose sugar. See here the structure of this ribose sugar and deoxyribose sugar. The numbering is the sugars carbon numbers are by 1 dash, 2 dash, 3 dash, 4 dash and 5 dash. The carbon number 3 dash, this one the carbon number 3 dash carbon. This is the 5 dash carbon. In this D-ribose sugar, there is also the numbering is the 1 dash, 2 dash, 3 dash, 4 dash and 5 dash. In this way, the 5-membered ring monosaccharides are present in the DNA and RNA. DNA containing the 2-oxyribose sugar and RNA containing the D-ribose sugar. Now see the nitrogenous bases which are present in the nucleotides. They are divided into two groups, pyrimidines and purines. The pyrimidines which are made up of the 6-membered ring. This type of the ring, 6-membered ring is the pyrimidine rings. The base present in this type are the thymine and cytosine. And second type of the base is the purines. This type of the base made up of the 6-membered ring which is fused with 5-membered ring. This type of the ring is called as the purines. See here 6-membered ring and which is fused with the 5-membered ring. And this type of the base containing the adenine and guanine. That means the pyrimidines base are the two types, thymine and cytosine, and purines base are the adenine and guanine. And these rings are not only made of the carbon, there is the presence of carbon and nitrogen. These rings are made up of the carbon and nitrogen. Only we understand here that the pyrimidines, that means that there is the 6-membered ring which containing carbon and nitrogen atoms. And in purines there is the 6-membered ring which is fused with this 5-membered ring. And they containing the carbon and nitrogen atoms. Then see what are the pyrimidines base. These are the structures of this pyrimidine base. The pyrimidine base are the cytosine, then thymine and uracil. And purines base are the adenine and guanine. The DNA containing the cytosine and thymine pyrimidine base and uracil which is present in the RNA. This type of the base is the pyrimidine base. That means there are the 3 pyrimidine base and 2 purines base. The pyrimidine base are the cytosine, thymine and uracil. And purines are the adenine and guanine. That means nucleic acids containing the 5 bases, 3 pyrimidines and 2 purines. Now see the nucleosides. The nucleosides are formed by joining the anomeric carbon of the furanose with nitrogen of a base. How the nucleosides are prepared or formed? The nucleosides are formed by joining the anomeric carbon of the furanose with nitrogen of a base. This is the anomeric carbon atom of the furanose. Carbon number 1 is the anomeric carbon atom, and it is linked with this nitrogen atom of the base. And that means sugars are linked with the base is called as the nucleosides. See here, this type of the base is the purines base, and this type of the base is the pyrimidine base. When this sugar is linked with the purine, then it is called as the purine nucleosides. And when the sugar is linked with the pyrimidine base, then it is called as the pyrimidine nucleosides. Purine nucleosides and pyramid in nucleosides. It depends on the base, type of the base which are linked with the anomeric carbon atom. This is the anomeric carbon atom of the sugar, furanose. See here, this is the anomeric carbon atom, and linkage between is the anomeric carbon atom and nitrogen base, nitrogen atom of the base. In this way, nucleosides are formed by joining the anomeric carbon atom of furanose with nitrogen of a base. Now see the nucleotides. The nucleotides containing the three parts, we studied this, that these nucleotides are the monophosphates of nucleosides. See here, nucleotides are monophosphates of nucleosides. We understand here that the nucleosides are the sugar and base. This is called as the nucleosides, and when these nucleosides are linked with this phosphate group, nucleosides are linked with phosphate group, then it is called as the nucleotides. The nucleotides are formed by adding the phosphate group to the 5-OH of the nucleosides. See here, these sugars having the numbering 1-, 2-, 3-, 4- and 5-. And this phosphate group is attached to the 5- carbon atom of this sugar. That means the nucleotides are formed by adding the phosphate group to the 5-OH of the nucleoside. And this type of the linkage is called as the nucleotides. In this structure, the base are the adenine, guanine, cytosine or thymine. And these bases are present in the DNA. In this structure, this nucleotide is the DNA structure, DNA nucleotide. And in this, the here presence of uracil. And uracil present in the RNA, and therefore this type of the nucleotides are the RNA. That means there are the two different nucleotides. This type of the nucleotides are present in the DNA, and this type of the nucleotides present in the RNA. Understand? The nucleotides are the phosphate group of 5-OH group of the nucleosides. That means it is the monophosphates of the nucleosides. This part is the nucleosides, and it is linked by this phosphate group. Therefore, nucleotides are the monophosphates of nucleosides. Now see the primary structure. The nucleotides consist of a linear sequence of nucleotides. Linear sequence of nucleotides that are linked together by phosphodiester bond. Phosphodiester bond. These nucleotides are linked together by phosphodiester bond. These nucleotides consist of the three components, which are the nitrogenous base, 5 carbon sugar, and one or more phosphate groups. See here, this type of these are nucleotides. This nucleotides containing the base. Then this is sugar, and this is the phosphate group. And they are linked with each other by phosphodiester group. Ester group is the oxygen. This one. There is the two ester group, two oxygen atoms here. See here. In this way, these two nucleotides, this one unit, and this is the second unit. These are linked with each other by diester bond. Therefore, this linkage is called as the phosphodiester bonds. We know that here the two nucleotides are linked together by phosphodiester bond. This one. Now see the formation of polynucleotides. Formation of polynucleotides. We know here these nucleotides are linked with each other by this phosphodiester group. The linkage, see here, the linkage take place. This one is the 5' end, because this phosphate group is attached to the 5' carbon atom of the sugar. This one, this is the 5' and this is the 3'. There is the linkage at the 5' carbon atom and 3' carbon atom. When this phosphate group is attached to this 5' carbon atom, therefore it is the 5' end, and in this way each nucleotides are linked each other by this phosphodiester linkage. And this one, see here, this is the 3' OH, therefore it is the 3' end. That means the polynucleotides having the 5' end and 3' end. 5' end, that means the carbon atom 5' carbon atom linked with this phosphate group, and 3' carbon atom is linked to the phosphate group. There are the two linkage of the phosphate group in this way. The two nucleotides are linked with this phosphate group at the 3' position. One 5' and one 3'. In this way, there is the formation of polynucleotides take place, and they are linked with each other by phosphodiester linkage. Now see the double helix structure of DNA. Double helix structure. The James Watson and Francis Crick put double helix model for DNA. This is the double helix model. This DNA consists of two polynucleotide strands with which wind into a right-handed double helix. There are the two polynucleotides, these two, and they are wind with at a right-handed double helix. They are wind into the right-handed double helix structure. These two strands run in the opposite direction. That means one strand from the 3' position and second strand from the 5' position. This one. These two polynucleotides are the different. These two strands run from the opposite side. This one is the 3' which is end with the 5', and second strand is 5' and which ends with the 3'. In this way, these two strands run in the opposite direction. The sugar phosphate backbone lie on the outside of the helix. The sugar phosphate backbone lie on the outside of this helix, and the bases lie on the inside of the helix, which are perpendicular to the axis of helix. These are perpendicular to the axis of this helix. See here, this is the backbone. The sugar phosphates backbone on the outside of the helix, and phosphate and bases lie inside the helix. The double helix is stabilized by hydrogen bonding between the bases of two strands. See here, these bases are on the inside of the helix, and these two bases are bonded with each other by hydrogen bonding. See this, all these bases are bonded with this hydrogen bonding. See, and these two strands of the double helix complementary to each other. In this way, the DNA have the double helix structure. Now see the complementary base pairs. The bonding between these two bases by hydrogen is called as the hydrogen bonding, and the complementary base pairs are guanine forms. This guanine form the bond with this cytosine. There is the formation of 3 hydrogen bond between the cytosine and guanine, and therefore guanine and cytosine are the complementary base pair. Then second is the adenine always forms the 2 hydrogen bonds. Adenine always form 2 hydrogen bond with the thymine. Adenine form the 2 hydrogen bond with the thymine, and therefore it is the complementary base pair, which is the adenine and thymine is the complementary base pair, and guanine and cytosine are the complementary base pairs, which having the 3 hydrogen bonds, and thymine and adenine form the 2 hydrogen bonds. Therefore, the adenine and thymine, C and G, that means cytosine and guanine, are the complementary base pairs. In this way, we understand here the double helix structure of DNA and complementary base pairs. Thank you.